Grand Rounds. I'm delighted to introduce today's speaker, Dr. Bony De Kumar. And Bony, it's a relatively new faculty in the Department of Genetics, and recruiting joined the year 2021, with the middle of the COVID. And more kind of many people know him. I was just kind of talking to him. I said he's the go-to person. He's the director for operation for YCGA. And many of you here have using either clinical or research for YCGA, I'm pretty sure. So I usually say, okay, he's the go-to person, get a lot of detail, know how you get your experiment done, and at YCGA, definitely it's a person use that and contact him multiple times. So Bony, before he joined Yale, he was the director of COBRA program, genomics core at University of North Dakota, and before that, he was at Bar Harbor Jackson Lab Genomics Laboratory. So he accumulated a lot of experience and for the genomics technology and the application, and he published extensively in the genome technology, also genome application. So then his rich experience and for both sides, so the technology and application make him very, very unique to the YCGA and to this community. So I'm very looking forward to hear what update from YCGA for the new technology and in the future. So Bony, take your floor.
Thank you so much. It's really my pleasure to talk in this forum. So I'm going to actually, the outline of my talk, I'm basically going to talk more about two aspects of recent development. One is cell-free DNA, and many of you are familiar with this, but I will be focusing my talk a little more extensively on the non-genic assays from cell-free DNA. So many of you are aware of using cell-free DNA for variant analysis, but I'm going to focus a little bit more on the non-genic assays using cell-free DNA and what actually biology behind those non-genic assays. Then I will be ending my talk with spatial technologies for new generation pathology. So we are going to see a lot of change in the spatial technologies, especially in this year 2023. So I'm going to give you a glimpse of what's going to be available for you to in this area. And the basic learning objective for this talk is to have a view or a bird's eye view of current and future applications of cell-free DNA in diagnosis. And also we will be learning about the spatial technology and how the future of this technology is going to shape up for pathology as well as diagnosis. So everything starts with history. There is a history behind everything that we do. So when we think about cell-free DNA, I heard this term very recently in 2017 when I was at Jackson Lab. And I think that many of us might have heard this word around that time only. And one of the reasons why that is a time when we started having sequencing became cheaper, and we started doing more and more with sequencing technologies. But the discovery of cell-free DNA is not that late. I mean, it's not that recent. The first time cell-free DNA was discovered way earlier, like in 1948, when Mendel and they actually identified that there are free DNAs that is circulating in the blood, and that has, so they don't know what is the significance of the circulating DNA, but they could actually isolate DNA from free circulating DNA from the blood. But real breakthrough came in 1977 when people realized that there is a high level of cfDNA in plasma from the cancer patient. So there is some kind of a relationship between a cancer patient and presence of high level of circulating DNA. And in 1989, first circulating tumor DNA was characterized. And actually based on that, then there were, since 1998, various aspects of or various characters of cfDNA were identified, like fragment size, what is the relationship between fragment size and a disease condition, whether the fragments are random or they are specific, and we are going to see some of those properties. So we have two sides of development in cell-free DNA. One was, which I termed as non-genic aspect of cell-free DNA assay, where we are looking at methylome, we are looking at fragment size distribution to understand about disease progression or tissue of origin and those kind of questions. And meanwhile, we are also looking at variants in somatic variants as well as germline variants to identify a disease condition. Like, as you see, many of those breakthroughs came in oncology, and we are going to see some other examples also from other diseases, where you can see mutation in particular certain genes were characterized using cell-free DNA assay. So cell-free. So currently, if you think about, so what is the origin of cell-free DNA? Where actually, or what kind of cell-free DNAs are there in our body? So we have what we call as cell-free DNA. It consists of 5 different kinds of fragments, DNA fragments. First one is circular mitochondrial DNA. Second is linear mitochondrial DNA. Third is single-stranded DNA. Fourth is double-stranded DNA. And fifth is extrachromosomal circular DNA. And many of you are aware that each of these components, mitochondrial DNA, extrachromosomal circular DNA, each of them have shown to have a very important role in disease as well as in diagnosis. So now, if you think about the origin, how this DNA really comes to bloodstream, or why we see this DNA into bloodstream, this is through actually multiple mechanisms. So, like if you think about apoptosis, the cells goes to apoptosis, and that releases nucleosome. So those nucleosome, broken nucleosome, mono, di, and trinucleosome, they are released into bloodstream. Similarly, necropsis or autophagy and all these processes leads to release of mitochondrial DNA or long fragment of cfDNA. So many times when we think about cfDNA, we think about their short fragment, but sometimes these fragments can be as long as 30 kb. So people have shown that there can be bigger cell-free DNA. And so source of those long cell-free DNA or mitochondrial DNA is basically through many of these processes. Now, there are some DNA actually, which is associated with proteins also. They also comes from some of this mechanism like erythroblast enucleation or netosis. Netosis is again a mechanism that is shown by, I believe it's some immune cells. They actually rupture and form a net of DNA when they are actually they are doing their function. So this process also releases a lot of DNA into the blood. And then many DNA protein complexes are also released, as including exosomes that are actually released into DNA. So there are multiple mechanisms. There can be apoptosis, there can be necrosis, there can be netosis, or even just the vesicles. As a vesicle which is encapsulating DNA is actually released into the blood, and all this DNA circulate within our bloodstream as a free DNA, not within the cell or a protected structure. So there is a word that is used for using cfDNA, and the word that is used called as liquid biopsy. The reason why this is called as liquid biopsy, that you are not getting into an organ to get a biopsy, rather than you are using just blood to get a sense of what those what is happening to the organ. So as per the NCI dictionary, this is a term that basically defined for cancer, but this is true for all other disease, all other tissues. So basically it says a laboratory test done on a sample of blood, urine, or any other body fluid. So basically important thing is that this test is done on body fluid. And what it actually recognizes, or it actually tries to detect small pieces of DNA or RNA or any other molecule released by this tissue, and in this context, tumor. So basically, by looking at whatever freely available DNA, RNA molecules, or other micromolecules present in our body fluid, you can actually come to a conclusion regarding disease, residual disease, minimal residual disease, or relapse, or even the stage of the disease. So that is called as liquid biopsy. There are 3 major areas where liquid biopsies are predominantly advanced fairly, and currently there are many clinical or the commercial level services are offered. The one is detection of fetal DNA in maternal blood. So this is one area where using mother's blood without getting into invasive procedures like amniocentesis or any other invasive procedure which actually which may harm fetus. Without that, you can just take a blood sample from mother and you can find out the status of fetus in the mother. Similarly, you can actually use cancer ctDNA or circulating tumor DNA in cancer patients for looking at metastatic stage, tissue of origin, or minimal residual disease, or even the cancer. Like what you can actually come up with a prognostic determination based on the ctDNA and the variant present in that. Then the third major area where cfDNA is used is the detection of donor-derived DNA in plasma of transplant patients. So transplant rejection is another area where there are assays available which is looking at increase in the donor-derived DNA in the plasma of the recipient, and that actually acts as an indication for transplant rejection. So these are some of the assays that is currently available in market. So as I was saying, so most of the cfDNA technology you can use for early detection of a disease, like case of cancer. You can use this for screening or early diagnosis. You can use cfDNA-based profile for molecular profiling. You can use this for minimal residual disease monitoring. You can also use cfDNA-based approaches for assessing treatment responses, or even you can use it for tissue of origin. So the advantage is that every time a patient visits a doctor, they always, it's easy to get a sample of blood. It's not like any other invasive procedure or imaging technology where it needs a separate appointment, or even for a consequence in terms of financial consequence in terms of insurance payment. But so we always collect blood for various purposes, and the same blood sample can be used for profiling various aspects of the disease progression. So that makes this technology very interesting. And if you think about the market share of the technology, currently in 2022, the cfDNA-based technology stands at around 4.9 billion in market share, which is going to increase as per some of the projection, going to show around 21.7% increase, and going to be a market share of around 31.1 billion in 2027. And one of the reasons why the major driver for this market increase will be, first is that there is a general consensus that rising cancer prevalence is on rise. Then people are focusing on early detection of cancer, and cfDNA-based technologies are making that possible. Then NIPT, or the prenatal diagnostics, are actually getting successful, and this success is actually is currently shown in public program, and I'm going to show you one such public program that is actually using NIPT using cfDNA as a platform. And also there is an increase in the R&D program. So a lot of cfDNA-based grant, even if you go to NIH, there are exclusive liquid biopsy grants available in NIH. So one of the major market players, as you can see, these are some of those very familiar names like Natera, Illumina, and Exact Biosciences, Grail. So most of the big players in the genomics market, they are into this business, and they are coming up with more and more kits for either extraction of cfDNA, library preparation, or the characterization of. So now, as I said, because of the success of cfDNA-based technology, especially in NIPT, California came up with a program called as California Prenatal Screening Program. So this is a statewide program. Every single pregnant woman will be screened for prenatal. They will undergo prenatal screening, and they will be given two kinds of screening. One is a cell-free DNA-based screening, and other is MSA-FP, regular MSA-FP screening. So this method, actually, which is a cell-free DNA-based method they're using, they will report for 4 diseases. One is Down syndrome, trisomy 18, trisomy 13, and neural tube defect. But this assay, what is actually California State Health Prenatal Screening Program is using, it can actually detect almost 24 different prenatal conditions, and which includes partial duplication, deletions, aneuploidy status of almost all autosomes, sex chromosome abnormalities, common chromosomal conditions, and so on. So almost like you can actually detect almost 24 different programs. And this is actually, this product is already approved in Europe, but it's actually not approved yet in FDA by US FDA, but this is actually available for other people also to implement, and Yale Health System, as well as New Haven Hospital, if they are interested, we are also happy to help you to establish such a system within our system. So as you can see, the success of NIPT using cell-free DNA also reflected in public policy perception, and public health programs started using this as a technology. So this is no more a technology that is more like in academic spheres, but really at the commercial sphere, as well as in the public health stream, it's becoming a mainstream diagnostic tools. So, but what is the actually main challenges of cfDNA technology? Though I have spoken so much about the field growth of this field and all this is going to happen, but there are really some important challenges this field is facing at this point. So one of the biggest issue is purity in extraction. How can we get a very highly pure cfDNA? And one of the main problem with this extraction is that it also includes, when you say cfDNA, it also includes exosomal DNA. So if in strict academic terms, they are two different things. cfDNA and exosomal DNA is two different things. So if you want to separate these two, exosomal DNA and cfDNA, there are methods available which uses microfluidics, but normal extraction process protocols, whatever they are calling as cfDNA, is basically a mixture of cfDNA and exosomal DNA. So this is one aspect, and we also always have to keep that in mind. Second, the way you handle blood. If you don't handle blood or any liquid, blood also have some cells, and there is a chance that those cells will rupture. And if those cells will rupture and release their genomic DNA into this fluid, then that will lead to contamination of genomic DNA. So handling of blood at the level of collection site, as well as during the pressure of cfDNA, is actually pretty crucial to prevent such a contamination. Second aspect is stability. So cfDNA, because being a small naked DNA, it's actually prone to action of nucleases. So that means you have to really stabilize those DNA. And specifically from urine, because it consists of large amount of urea, you will see a rapid degradation of cfDNA when you collect it from urine. So one of the suggestions or one of the recommendations from YCGA based on our experience is that we should always collect blood. If you are going for cfDNA extraction, collect them in Shrek tubes. So there are tubes that are actually meant for this purpose. So you can use Shrek tubes, and that is the tube that you should collect rather than using normal blood collection tube. So far as urine is concerned, collect urine and transfer them into ice as quick as possible, and then extract cfDNA that day. Because if you go for next day, we have seen a huge degradation of urine. Most of the time, we cannot actually even recover cfDNA from urine if we go for next day. If you want to freeze your blood, freeze them as serum, because we have seen a high purity of cfDNA from the frozen serum compared to even the fresh blood. The third major challenge is that even if whatever amount of you use whatever amount of starting material like urine, saliva, blood use, you will get a very low yield of cfDNA, except for the cancer kind of situation where you have a huge influx of the cfDNA in blood. So we have to come up with a library preparation technique that can handle low yield input. And similarly, the other major problem is that among all the cfDNA, the major cfDNA contributor is blood cell itself. And then whatever cfDNA is contributed from your organ of interest is actually a very low percentage. It's less than 1% of the cfDNA is contributed by your organ of interest. So if you are looking at, so that means you are looking at a situation where you are analyzing low allelic frequency variants. And in that case, you need a robust method that can actually call true positive and true negative. So here is the example that in YCGA, we have optimized protocols for extraction of cfDNA. And as you can see, this is a profile of blood as well as serum cfDNA. And blood, we could actually, this particular sample has almost 86% purity of cfDNA. And this serum sample has almost 95% purity in cfDNA. So we also optimize protocols for isolation of highly pure cfDNA from CSF and urine. And we are also trying currently to look into plasma as a source for cfDNA and see that whether we can isolate a good quality of cfDNA from plasma. So now at this stage where we are at YCGA, we can isolate cfDNA from blood, urine, serum, and plasma, as well as from the CSF. And so if your sample belongs to any of this category, we are happy to help you with the highly pure cfDNA. Now, I told you like there is a issue of low frequency alleles because actually only 1% or less than 1% of cfDNA is contributed from the tissue. For that, we are using a different methods of prepping library called as duplex sequencing. In duplex sequencing, in simple word, what happens that you are adding a randomized duplex tags on the plus and minus strand. So basically, you have a unique UMI on your plus strand and another UMI on your minus strand. So when you merge these reads together, then you need to see same variant on your plus and minus strand both. So like in this pictorial depiction, you can see this green one. Green one is present in all plus and minus strand, corresponding strand. But this purple one or this blue one, this blue one is actually randomly present. So we know that this is only present in one strand. So this is a PCR duplicate. So by looking at this, we know that, okay, other than this green variant or green change, this is the only true mutation. On all other mutations are actually either randomly created during sequencing or using or during the PCR cycling. So based on this, even the frequency of allele is very low. We can detect those low allele, frequency alleles also with very high level of confidence. So we have implemented duplex sequencing within the cfDNA sequencing platform. And so now our all sequencing preps for the cfDNA is through the duplex sequencing library prep. So we have taken care of the purity of cfDNA, challenges regarding purity of cfDNA. We have taken care of the issue with the true positive with the low allelic frequency variants using duplex PCR. And we have protocols now for generating library for variant calling, whole genome sequencing, targeted methylome, and whole genome methylome for as low as 6 nanograms of material. So if we have 6 nanograms of material, we can prep any of this kind of libraries. So whole genome sequencing, targeted sequencing, whole methylome, targeted methylome sequences. So with that, as I said, like all these 3 problems, like low yield, we have taken care of that using a very specialized techniques. This one we use duplex sequencing, and then the purity, we now know that the protocols are optimal to get highly pure cfDNA. So let's think about what are the uses of cfDNA. So one of the major use when you immediately think about cfDNA is looking at the genomic variants in the cfDNA to identify a particular disease condition. And this was an obvious, very obvious application. And as you can see, the first few of the applications that actually came off of cfDNA was from looking at Y chromosome of male fetus in maternal blood, or various kind of alterations in tumor and non-tumor patients, like looking at microsatellite length or chromosomal alterations, somatic variants, and all. So these are actually all based on variants and variant analysis, and also looking at SNP variations in donor and recipient for figuring out transplant rejection. So these were some of those earliest methods, and all of them were based on looking at variants in cfDNA to identify variants. So here is one example where we have used variants, and the difference between us and others is that we were actually using just exomic regions. So we did a targeted capture of exomes, so like from every single gene, 22,000 genes, and then we looked at exomes, variants in the exomes in the 4 different kind of materials from the same patient. So this is a pineal glioblastoma patient, and here we have so we have used CSF from the same patient. We use blood plasma, we use genomic DNA, and we use tumor DNA for looking at variants. So we did a targeted capture, we sequenced it, and we looked at the variants. So as you can see, a large number of variants are common between all these 4 material. But we are actually seeing a higher number of variants called on this one. So we are still checking up why we are getting higher number of from cfDNA. And one of the explanation at this point is that we are actually sequencing slightly deeper for cfDNAs because we don't have a way to say how the coverage needs to be calculated, because the coverage is based on whole genome length. And since this DNA, we don't have that much, like what other regions will be coming into cfDNA, we cannot have a priori knowledge of it. So we are probably sequencing slightly higher, and that is what giving us more number of variants. But the take-home message is that the large number of variants that we are seeing in genomic DNA, so which is basically germline and somatic variants that we are seeing in germline variants and somatic variants, are also present in CSF as well as in blood plasma. So this is kind of gives us more confidence to look at this as a way. But we went a little more further. So instead of actually focusing on whole exome, where from a clinical perspective, it is very difficult to build them as a whole exome assay. What we did, we actually decided to do a targeted enrichment of cancer-specific gene. For that, we used a probe set called as GOAL panel. So what is GOAL actually? So GOAL stands for Genomic Organization of Academic Laboratories. It consists of 28 members, and Yale is one of the members of this organization. So what they do, they generate panels and they manufacture them and they divide them among the cooperating laboratories. So this way, the panels will get cheaper in terms of price, and then and those genes can be easily validated. The good thing is that people actually talk to each other, they get protocols, they exchange ideas, they help in troubleshooting each other. And also this is a platform for molecular testing in academic setting. So that gives a lot of flexibility in working with various labs with various kind of expertise. The future of this organization is basically going into more like a concordant studies, as well as providing data sharing resources, as well as multi-institutional studies, and also expanding the network, and including more number of institutions. So GOAL, as you can see, I have just tried to make a rough outline of USA with this gene names, but I don't know whether it is obvious for many of you, but I just thought it's a creative way of putting genes there. So this GOAL panel consists of 527 genes, and those genes are important for cancer. So some of the genes that are shown as an outline of this US map is basically those are the genes that are MCG actually is actually calling this as a reportable gene. So that means you can actually get billing for this gene. So you can actually, insurance agencies can bill, you can bill insurance agencies for these genes. The good part of the GOAL panel is that many of investigators and clinicians in Yale health system, they are aware of this panel, and currently they are using it for heme tumor profiling. This panel also includes genes or regions from mitochondria, as well as translocation, some of the cancer-specific translocations also in this one. So this is a very short panel, but it's actually very powerful with a lot of information available for us to work with. So we looked at the status of those 527 gene and the variants within. So for this particular region, we captured the exomes of 527 genes and a mitochondrial region, and we sequenced it at a very high depth of 500x, minimum 500x coverage. And so we did that same for CSF, blood plasma, as well as genomic regions, as well as tumor DNA. And we found that, again, like most of the somatic and germline variants that we can identify from genomic and tumor DNA, we could actually identify that in the cfDNA, either in the CSF or in the blood plasma. So this shows the power of this method, that we are actually able to identify somatic variants as well as genomic variants in the cell-free DNA, irrespective of source, like either CSF or blood plasma. So this is something, this was one of the very obvious application of cfDNA when cfDNA field was evolving. But very recently, or well, rather say last 20 years, there are many assays that are based on non-genic features of cfDNA. So which basically not something based on variants or some those kind of. So what are the non-genic features which are actually used for characterizing cell-free DNA? So one of the features that actually frequently used is cfDNA methylation pattern. The other one is fragment size. Third is end motif frequency. And we'll see some of those examples as we go. Jagged end length, preferred end coordinates, orientation and density, motif discoveries like within the end. And there's another something based on the periodicity of the fragment that we see called as window protection score, and cfDNA integrity, and nucleosomal footprinting. So these are non-genic features of cfDNA that is employed for diagnostic purposes, and we are going to see some examples as we go. So one of the earliest example of using cfDNA methylation for a marker was actually identification of hypermethylation of collagen type 12, which seems to be increased in metal blood with a high blood pressure condition. So this was one of the first marker, was a methylation marker that identified as a cell-free DNA marker for in a disease condition. Then it was also shown that the DNA methylation pattern of placenta and the cell DNA that is originating from placenta during gestation have a very characteristic signature when mother is going through a gestational diabetes. And then finally, it was identified that there are two hypomethylated markers which were considered as a universal fetal marker, which was SERPIN-P5 and RAS-F1. So these two, one hypomethylated marker, these two markers were actually still now, actually, it is used as a quality control and quantification marker for fetal diagnosis. So basically, the level of their hypomethylation is considered as a constant, and then everything else is calculated based on that. And and so it's still used. And then finally, the cfDNA methylation has a very important application in finding tissue of origin. So especially cancer, where highly metastatic cancers, they want to find out where this old cancer originated, what is the primary site of cancer origin and all. It can be actually done with the help of methylation, cfDNA methylation. So, but there are certain challenges that has when we are using cfDNA methylation profile. So one of the biggest challenges is that different tissue exhibit different epigenetic modification or methylation pattern in a body. Now, each of these methylation patterns of cell-wise or tissue-wise is not actually completely worked out. So that's the biggest challenge. Now, the second thing is that these modifications, especially the methylation, this changes according to age, diet, and then lifestyle. So that means, like, you have to really, and also gender is one of the biggest determinant of modification, differences in the methylation pattern. So that means you have to create a whole repertoire of methylation pattern before you start using it effectively. Then, till recently, we used to need a large amount of starting material for analysis. So to make a library, we need at least 100 to 200 nanograms of material. But as I said, like YCGA and others have now protocol where we can use as low as 6 nanograms of DNA, and we can still make library out of this. And then finally, the cfDNA is a mixture of various, so it's a mixture of DNA coming from various tissues. So that means you need advanced statistical analysis tool to deconvolute this data to find the origin of each of the cfDNA. So thinking about these challenges, but then still we have a lot of promises in this area. So how this is done? Basically, you will collect the blood and then we isolate cfDNA, and DNA is treated, which is basically conversion of your methylated or non-methylated residue into a different nucleotide. And we at YCGA, we have both protocols like bisulfite sequencing and enzymatic conversion for these purposes. So you can use either of them. And once you have this, you make a library, sequence it, and data will be analyzed. So this is actually the flow. It's like any other NGS protocol. The only difference is that you will starting with the cfDNA, and again, the material that you have in your hand will be very less. So I will just show you one small example beyond cancer where we have used methylome analysis of cfDNA in PKD patients. So it's a PKD stands for polycystic kidney disease. And we compared two different sources of cfDNA. One was urine and another was blood. And we were actually looking at whether are we seeing same kind of cfDNA between urine and blood. So as you can see, there are, at first look, it doesn't seem like we have many of those cfDNA or methylated region that are similar between these two, because we are seeing a smaller number of fragments from blood and higher number of fragments from urine. And one of the reasons why that is happening is, as I mentioned, that urine consists of very high level of urea that actually leads to further breakdown of cell-free DNA beyond whatever actually we are seeing initial breakdown. So this is a general distribution of number of different loci that we saw under different conditions. So in this, we could actually find at least 6 of the loci that are unique to kidney-specific gene expression, and we are going to see some of the genes as we go. And then we also saw, we identified around 2,900 or around 3,000 microRNAs, microRNA-related regions in urine, as well as 773 in blood, in which 195 regions are common between these two types. But if you look at real overlap between these two, irrespective of their methylation ratio of methylated versus unmethylated unmethylated base, which is called as beta value. If you actually, irrespective of that, if you are looking at, then you will see there is a huge overlap between blood as well as urine. So there are only very few unique blood-specific cfDNAs there in this sample compared to the common ones. But when you actually set up a threshold, you will see this overlap actually reduces significantly, the percentage of overlapping regions. So if you're looking at least 20% regions that have methylation, and in that case, you will see drastic loss of that overlap. So one of the reason, biological reason, it seems, again, has to do with the environment in which urine is. So some of the alkaline nature, as well as the presence of urea, probably is leading to a demethylation. So we think that is what's happening in this case, but we are not yet sure. And many literature are actually indicative of it, but nobody have ever tested that hypothesis in a concrete manner. So now we looked at what are the genes, those which are near to the differentially methylated region. So the nearest neighbor gene of differentially methylated region, and here we are showing around 6,000 loci in the urine, as well as 1,921 loci in the blood, which is near to the gene that are expressed some or other way near kidney. So, and you are seeing some of those top expressed genes. These are like PAX2. Some of these genes are actually very specific, SLC17A3. These genes are very specific to kidney. And we are seeing a differential methylation of those regions controlled to patient versus control sample, and those are near to the kidney. Here is a list of those microRNA. As you can see, many microRNAs are also upregulated or hypermethylated or hypomethylated in blood and urine. So we have some of the very interesting candidate to further work on. We haven't done much of the biology here because our purpose was not to dwell into the biology of it, rather than we were optimizing our pipeline. So that is where I'm just showing you some very superficial data on this. But what I would like to actually emphasize here is that we could identify differentially methylated regions which are near to kidney-specific genes. And we are also able to identify a large number of microRNA, which could potentially act as a biomarker if the study actually taken further. So finally, we could actually nail down to 10 regions or 10 genes which has nearby regions with differentially methylated region. And these are those genes. So SLC17A3, SLC5A12, PROS, SLC5A11, FTCD, PAX2, NXPS2, KCP, PLOT5, and OGDH. So these genes we identified which have a differentially methylated region nearby when you compare between patient and the control samples. So I think I was able to show you that you can use a very small amount of input sample and can do a methylation studies using cfDNA. But other exciting aspect, I have shown that there are many unique characters of cfDNA fragments, and you can use this as a means to identify various aspects of disease and diagnosis. So there is a model size is there, which is actually around 166 base pair. This is model size distribution of cfDNA is 166 base pair. A 10 base pair of successive peaks you can see after 143 base pair. This is called as a periodicity, and where it's coming from? It's coming from the nucleosomal organization. So DNA is wrapped around nucleosome core, and that wrapping is around 140 base pair. So maybe the first two bases either side is protected, and after that it cleaves at a 10 base pair periodicity based on the pitch of the DNA. So basically the one helical turn is around 110.4 base periodicity. So it will go and cleave at that. And that's where you are seeing that 10 nucleotide periodicity in this. And then there are some other characters like fetal DNA is always shorter than the mother's DNA. And this again leads to the nucleosome organization and the chromosome accessibility. And then open chromatin always sheds more cell-free DNA. So you can use this as a marker for looking at cell-free open chromatin. Also, it has a very characteristic. So all the cutting and the chipping of cell-free DNA is actually not random. It's actually at a very specific position. And that's why you can use this as a marker for characterizing various pathologies, especially tissue of origin. And cancer cfDNA is also always associated with shorter length. Again, that is because of loss of nucleosome from the many of the genes. And it's not a random, process. So as I was showing like this, all the processes can actually, I'll just skip this slide. So this just shows you like what are the, where are this coming from? So anything that actually outside of this nucleosomal core, that gives you this 10 base pair periodicity. Nucleosomal core actually gives you 145 base pair fragment, which is basically the whole nucleosomal core. 167, which is present as a predominant fragment, is one which consists of nucleosome core plus H1 linker histone, and sometimes you have whole complex of H1 linker histone along with the histone chromosome that will give you 197 base pair fragment. And then you also see periodicity like dinucleotide, trinucleotide, and which will give you multiple of this 167. So basically you will get some around like 230 and like that. Also 330 and then so like that. So these fragments are very characteristics for tissue as well as for various conditions. So like in case of PKD disease, we looked at the control versus PKD, and this is the cohort 3 is control, and you can see in blood, you don't see much of the difference. So this cohort 1 and cohort 3 is from blood. So there you don't see much of the difference. But when you look at urine, the control fragment, which is cohort 4, has a model size of 166. So you don't see any other fragment size in the control DNA. But when it comes to patient, you will see a huge number of fragments coming from other fragment size. So this shows a characteristic difference in the control versus patient, and this can be used as an attribute for following up the disease condition. And sometimes you can use this for early detection of disease just by looking at fragment size, saying like if you are deviating, how much you are deviating from the model fragment size. So here is another example. So cohort 1 in this case have actually, so cohort 3 is the control here, and it is showing a very different profile than the cohort 1. And same thing as you can see here, cohort 2 is showing a different profile than cohort 4. So cohort 4 is again control. So the difference in the, and this is what we are looking at the periodicity that I was talking about, 10 base pair periodicity. So what actually it shows, like in the disease like PKD, which is a very clonal origin disease, where also you can actually use cfDNA as a means, or their fragment size distribution as a means to look at the disease situation. To conclude this part, we optimized automated cfDNA extraction using Kingfisher system. We have duplex library preparation, which we already optimized. We have already standardized the protocol for exome capture from cfDNA, so that is available. We validated gold probes. So as you have seen in case of pineal glioblastoma's case, we can do whole genome methylation using low input material, which is useful for cfDNA kind of. And currently we are working on targeted enrichment of methylome from gold region, and hopefully we will have some data soon on this. So we will have the panel that will look specifically into those genes, 527 genes, and the region around from the methylation perspective. So that will reduce cost of methylation analysis considerably. For this, I'm just actually moving towards my last part of the talk, which is spatial multiomics. The reason why I actually decided to include this, because if somebody asked me what I would like to call this year, 2023, so like you have Chinese New Year, rabbit, and lion, and those kind of names, I will, for being a genomics person, I would like to call year of 2023 as a year of spatial technologies. A swamp of technology is actually going to hit market in first quarter and second quarter of this year. So what actually spatial resolved single cell multiomics means? It means that you are looking at one tissue and you are simultaneously assaying gene expression as well as other epigenetic modifications, including proteome or metabolome on the same tissue, that is at single cell resolution or subcellular resolution, and including the spatial context of those those cells within the tissue. So you have spatial context, you have resolution at single molecule level, you have submolecular level, and you are also integrating multiple omics data sets into one single experiment, which is actually makes it a very powerful approach. So spatial transcriptomes can be actually divided into two different categories. One is sequencing based and second is imaging based. So one of the examples for imaging based is Murfish, which is offered by Visgene or Resolve. Then there are other technologies like NanoString or Xenium, NanoString Cosmex and Xenium from 10x that actually uses an image based approach. Then there are 10x is Visium, and NanoString has a platform called a GeoMax. These use a sequencing-based method for identification of transcript. So the question is, why spatial biology? So this can actually identify various types of cell within a tissue, even with whatever. So we can actually think about heterogeneity of cellular heterogeneity within a tissue type. It gives you information about location of cell in the tissue, whether is it deep inside the tissue, like a tumor, or is it like outside in the periphery? Because this have a consequence in terms of how much vascularization is there. So that also have implication in terms of relapse and other questions that people are asking. Then what are the biomarkers that are co-expressed, and what is their co-expression pattern? Then what is the cellular organization? Which cell is actually near to each other? What are the neighbors of those cells? And then finally, how this interaction, cellular interaction, is going to influence the microenvironment of the tissue. So you will have all these questions. If you have any of these questions in your mind and you want to answer them, that's where you need spatial biology for. Most of the spatial technologies, what they are doing, they are actually using multiple oligo probes against single gene, and each of those probes will be labeled with a dye. And then you actually enhance the signal from one molecule, and then you image them. This imaging part, like in case of each of the companies are using, or each of the technologies using different ways of doing it. So one such technology is called as Murfish, where they are using a combinatorial barcode. So they will have, so this region that will go and bind with your gene of interest, so hybridize with gene of interest. And then that probes will have a secondary hybridization region, which is actually when you add your secondary probe into it, this signal is amplified like what we are seeing here. And each of them, the amplification will add extra fluorescent molecule, and that will give you an extra glow of that site. And then you are using a combinatorial code. So like you usually go through a 16 round of hybridization. Like in this case, you can see if you have only hybridization at a probe is hybridizing, or color you are seeing at 1, 5, and 12th round of hybridization, then that means that particular spots belong to this gene, DYNC1H1. If you are seeing a hybridization signal at 4th round of hybridization, 7th round of hybridization, 10th round of hybridization, and 12th round of hybridization, that means you will see this gene, EGFR. So based on this combinatorics, you can identify a large number of genes in a single run of hybridization assay. Theoretical limit is actually 64,000. Because it's a 4-bit inscription. But since cell has only limited number of space, you cannot actually identify whole genome or every single gene in the genome in single cell. So ideal number is somewhere between 300 and 500. So which actually, without crowding effect, you can actually identify single dot and single, you can get that at single molecule resolution. So, but currently we at YCGA, and so I just want to show you how it looks like. So basically, if you amplified probes are hybridizing, you can see a single dot, which each of this dot pertain to one molecule of RNA. So it's a single molecule study. And currently people are, like as I was a postdoc, we have actually used some of this earliest method like Stellaris and HCR. And we use this dot for identification of nascent transcript as well as stable mRNA also. So now, we have a platform here at YCGA, which is called as Murfish or Seqfish. And currently we have a mouse panel and human panel with 100 genes of various signaling pathway. We also have a small neuro panel with 41 genes. And we are always happy if any one of you want to actually collaborate with us and develop your own panel, we are happy to do that. Currently, we are almost at the stage of working on final pipeline also for analysis. There's a company called as Resolve Biosciences. They came up with a platform called as molecular cartography. And this is again based on Murfish. The cost of this instrument is 400,000 and cost per sample is around 2,300 to 3,000. Minimum transcript length required for detection is around 500 nucleotide. Subcellular resolution is around 300 nanometer, and actually each dot is a single transcript, so you can measure. And after the processing, tissue remains intact, so you can use this tissue for anything else like you want to verify this for. The biggest problem with this instrument is that you need to process 8 samples simultaneously, and you cannot do less than that. Need optimization, and only 500 valid targets are available from this company currently. The other method is nanostring cosmex. This is a very powerful technique. The instrument cost is approximately $295. Again, the sample cost is something around $200 to $300. It gives you a better resolution at subcellular level. You will get less than 100 nanometer resolution. You can do simultaneously RNA and protein readout from this instrument. It is basically RNA, you can do up to 10 to 1,000 plex RNA, while 64 plex for protein is currently available. So it also comes with a informatics portal, which can call us Atomex. So you can actually use this for data analysis. So it has that flexibility that you don't have to work with a bioinformatician to analyze your data. This is another platform from 10x. Again, this is the imaging-based platform, but the probe that they use called as molecular inversion probes. So basic idea is that you have a probe, two probes hybridizing at the nearby location, and there is an extra region. And once you do a PCR, you will join this region, and then you amplify it. And that's how you identify your transcript of interest. And this is, again, this is going to be in market. It's already in the market now in the first quarter of January. There are other platforms like Akoya and Singular PX. This is also the platform that's going to be available in coming days. So what I just wanted to show you that there are multiple platforms that are going to be available in this year of 2023. And some of them we are evaluating for. And while we have already in-house Murfish for such platform, but we are also evaluating one or two platforms for buying them at some point. So the question that I actually this is a terminology that I coined myself, so it's not somebody big person's terminology, but I think this is a rational terminology in sense that where are we heading with? Are we heading towards a computational pathology? So what we mean by that? So if as a pathologist, you are interested in cellular phenotype, you are interested in cellular abundance, you are interested in localization of cells. And then you are also, if you get an ability to look into multi-omics data like proteomics, metabolomics, transcriptomics, or epigenetics data at the single cell level in a tissue in a spatial manner. And if you can integrate all this into a AI-based interface, and it can generate a digital report like the way we are doing currently for variant calling. So this is where the field is going. Some people are actually started calling this as a digital pathology. So this is what actually we are heading to. And then basically this means that we are going to combine spatial transcriptomics, we are going to combine spatial proteomics, all this together into single platform on a same tissue. And then we are training our AI programs with various parameters, and those parameters will give you a differential diagnosis, and those diagnoses can be used by a pathologist for reporting purpose as well as for the clinician to make clinical decision. So this is where actually currently the pathology is probably going towards with this invent of all these new platforms. So with that, I am not going to be going to give you any big conclusion, but what it seems like that many areas of diagnostic will be benefited from cfDNA based approaches. And as we are seeing that it's not just cancer or the neonatal, as in case of PKD disease, which is a very complicated disease, we are seeing that such an approach can be useful. We also tested this in stroke patients also. So we have some data on that too. So I think that there are many areas of diagnosis where this will be useful. And then anybody with our group or interested in developing something like this, please contact us and we're happy to help you with design and other things. And also, we are going to see in coming future, like we are going to see more inclusion of spatially resolved technologies as well as multi-omics approaches in future in pathology. So that is where the future of pathology is actually going to be. So this talk is just to prepare that what's going to come, and so hopefully we will be ready with our strategies. So just to finish this talk, I would like to acknowledge my YCGA family, especially Srikanth, who was driving force behind a lot of this work. Then Chris, Irina, and Grace were actually instrumental in optimizing most of the library prep extraction protocols. Chris and Lorraine, they were actually helped us in sequencing and putting them on onto sequencer. Jim, Francis, and Antrich, they were helpful in sequence data analysis and coming up with some of these pipelines. And all other members. So I haven't shown some of the other works done by our other members of YCGA for helping and optimizing various protocols, but all of their hard work is actually behind some of these presentations. And we have many collaborators who were who were actually instrumental in giving us samples. So like Lorraine Sansing, Carla Rocklin, Saurabh Ghosh, Stephen Somlo, Neera Dhal, Whitney Best. So all of this, our collaborator who gave their valuable samples to us to test. And above all, I actually, a big name I'm missing here is Kaya. Kaya is actually still actively involved in this process, and his valuable contribution is always there for us to guide us. With that, thank you so much for being here today and listening. Thank you.
Well, Bonnie, that's a wonderful talk and covered this new technology. So that's open for question, comments. Before that, if anyone want to see me, please and see the chat for the code number. So now anyone, I see the hand raised, and Mohammed, you go first.
Yeah, it was a great talk. My question related to the variant allele frequency in solid tumor. So you have, you said 6 nanogram will be enough, but we have solid tumor who shed DNA in the blood, and solid tumor, they are not that good in shedding. And when we have like oligometastasis, so in your experience, how did you encounter the difference in the cell-free DNA different by tumor type? And what's the variant allele frequency you can go to your threshold for detection?
So we haven't done that, but what I can tell you that we even made library from as low as which we cannot really quantify. So we know that there is a profile there, but when we are trying to do our quantification approaches, so that means it was less than 1 nanogram. So we tried in a couple of brain-related conditions. So what I can tell you that even with a very low, so the method that we are using, it can actually use as low as 10 picogram of DNA. So I don't want to actually advertise that, because tomorrow somebody will come and give us 10 picogram of DNA, and then we'll be in trouble, because as you reduce the quantity, chances of success is always keep decreasing. So but 6 nanogram definitely is something that we can always work with. So the first part of the question, that can we actually use even those conditions where tissue is shedding very low amount of cfDNA? Yes, the answer is even up to 10 picogram, which is present all the time, that we know for sure. But other question, we haven't actually tested that how low we can go with the allelic frequency. So we are currently, some of this data is just being generated, and then we want to really, so that we want to do with some of the cancer data, and we are going to get some samples from Murad's group. Kaya is actually trying to get this from there. And we are going to try some of those things, like how sensitive this, how low we can actually go with the frequency. And then probably the deeper sequencing can actually help. And that is why we were trying for the GOAL consortium probes panel for targeted enrichment. Then that means we have only 500 genes. And then if we go deeper sequencing of them, say 500 to 1000x sequencing. So we can do that at a reasonable cost compared to whole genome sequencing. And then that will increase our detection limit. So that is actually the final goal of this.
Dr. Bear.
Yeah, we're running out of time. I think my question's very targeted and short. When I might have missed this, but when you're talking about cell-free DNA from body fluids, like that aren't blood, you talked about urine, for example. How do you know where the DNA is actually coming from? Because if we look at urine, most of the cellular DNA in urine is probably from white blood cells, and those white blood cells are breaking down all the time. So cell-free DNA is also going to be from white blood cells. How do you know what you're getting your cell-free DNA from?
So that is what actually we are currently doing. We are actually running some deconvolution pipelines to see, based on methylation, that where this DNA is coming from. And you are right that most of the DNA, even in the blood comes from the blood cells. And same thing goes to urine, that the cells that actually predominate urine, they will give rise to a larger number of DNA. So that is where the real fraction of DNA that actually makes sense, or that is actually useful for us, is only less than 1%.
Okay.
Any other question? Yeah, we are at a time of the 10:15. Any other question? All right, so great. Thank you so much, Bonnie, for your great talk, and we definitely follow up for this kind of brand new technologies. Hoping we get everyone's interesting to collaborate with YCGA or any research or clinical project.
Thank you so much.
Thank you.